in Hawaii, and I don't think there are any over in the Marianas as well. So they're a Kusky.
Oh, thank you so much, Bruce. There are a whole bunch of fish in the crack here between these two gigantic boulders.
Yeah, so Bruce, I think this is Okeanos, this one that's coming into the center right now, but maybe I'll get your opinion too about this.
That's the big guy?
Yeah, the big red one that's just did a little bit of a circle there right in the center between the rocks.
A little bit of a circle.
Yeah, it gets confusing. There are two red fish in there. I think there's the brighter red Central Barracks and then the darker red Okeanos. You're right about that. There's one species, Okeanos japonicus.
He's coming up.
You saw the very long pelvic fins, the paired fins on the belly behind the head, and also long lobes on the dorsal and anal fins, the unpaired fins in the back, a sort of a rounded off caudal fin. If it turns around and comes out again, we can see those very large pelvic fins that are very characteristic.
Ouch.
This is in the family of fishes that are called glass eyes, Veo Veo in Hawaii. Okeanos is the deeper living one and the largest one. I think Chris Kelly actually has the state of Hawaii record for catching this species.
Well, there's the thing to
I'm not
grab
getting into the
tether
event
right there.
but we're seeing Okeanos, we're seeing Central
the rock by
Barracks,
your
we saw
skin.
an Odonanthius here.
On the right side.
You're
talking about the one ahead, yeah?
Not that it's anywhere near it.
Very rich site
Keep an eye out
for
for it.
fish diversity.
It's just an example.
Yeah, we agree, Bruce. This is an extremely rich site for fish diversity.
It's like, Carl, you seem awfully calm about
Probably
this.
more than we've seen any place else.
Yeah, I
on
think
any
so,
of our,
Shirley.
on any of
Yeah,
the flags.
absolutely.
Yeah.
I'm definitely feeling a bit more current now. Perfect. Josh is about to step in.
Mind if I zoom to lose the skids?
Go ahead. Yeah.
Looking to get your fix on swag, caffeine, or sugar? The ship store is now open for the next 10 minutes. As a reminder, the Call of Duty tournament sign-up ends 1700 tomorrow.
We're all laughing here because various times during the day, random times during the day, the ship store is open. And the person who operates the ship store is really funny. And so he always makes this hysterical announcement about different things that are different specials of the day.
Yeah,
and
his
so
monologue sounds a lot like, I don't know, more like Stephen Colbert than Johnny
really
Carson.
Funny. So we were all standing here laughing.
There's an awful lot of biology here, and it is incredibly interesting. The geological reasons for what you're seeing are so much larger scale. It's kind of hard to get a grip on it without seeing the region itself as a scale, at a scale of a map or something. But when the Pacific plate is subducted at the trench, it certainly goes down into the deeper mantle of the Earth, but it also has a vertical component of sinking that tends to pull it back eastward toward the Hawaiian Islands. So what happens then is that the overriding plate follows the trench. So all of the crust, the lithosphere, that's the crust of the Earth, and the hardened upper mantle of the Earth gets pulled apart. So there's a great deal of tension in this environment, and that splits portions of the seafloor apart from one another. We're looking at a seamount that sits right on the major fault that bounds a small ocean basin to the west of the island arc of the Mariana Chain. So very likely some of the fractures we're seeing that are producing these huge boulders that look like they've just been torn apart are a consequence of the fact that this volcano has built itself with magma oozing up along that major fracture that separates a 200-kilometer wide plate that is above the downgoing Pacific plate from the part of the seafloor that is being torn apart by this massive regional movement of the Pacific plate as it rolls back toward the east. Geology lesson for the day over. Over to you, Shirley.
Well, one geology lesson. I mean, there's lots of geology lessons here. I mean, again, this is just such a target-rich area biologically and geologically. Bruce was saying that it's impossible to log. Oh, no, that's Chris. Chris was saying to Bruce, it's impossible to log all of the species. So what we're doing is just concentrating on the ones that are new that we haven't logged before, because otherwise we're so busy kind of typing and entering things into the event log
and
that we're not looking. You just can't take your eyes off of the screen, off of the video. There's just so much. So many fish, and here in the like it looks like here the sponges now have been replaced by crinoids. So we've got lots of crinoids all along the ridge.
And you see crinoids on that little rock that the fish is just swimming by, the redfish, and I'm wondering if some of those were dislodged and dropped into that crevice during that more recent volcanic activity that was identified on this seamount.
Oh, so you mean like it would have picked it up and dropped it in?
Nope, I think the two rocks on either side just split apart, and that keystone block fell in.
Cool.
Yeah, and the crinoids may have survived or may have repopulated.
Yeah, and remember, because they can move. They can move. They can
That's
move.
true. That's true.
They can move.
Yes, I have you on survey 1.
But that certainly looks like a keystone drop block that just fell down from when those two pieces on either side of the screen parted.
Great.
These crinoids are everywhere.
Yeah, really, really common. Abundant. Yeah. Asako did a quick count. At least 75.
Asako's very good at this.
I know. At least 75, just in one screen area.
And Mary Jo is saying she's our petrologist on shore, and she's saying it's very surprising that there really are only two different types of lithology, two different types of rock types that we've seen on this dive. And perhaps just two really big eruptions rather than many small ones.
So
Thanks for that insight, Mary Jo.
Sonar started working again. Any idea what happened there?
Oh, look at the crinoids underneath that block.
Yeah, you just have to restart
yeah.
They're everywhere.
Right, so, I mean, there's just current that's flowing there, so they're able to
it.
feed.
Okay, great.
So, Navigator, what's our
and lots of tiny little fish.
Crinoid battles going on here.
Really, whatever the ridge
Crinoid
determine
battles.
who gets on top and who gets on the bottom.
Really?
The ones on the bottom, the ones that kind of lost the fight.
Oh, could be.
You never know. They may be getting even more food down there.
That's what I'm thinking,
never
because cracks like that are going to force the current to go faster, right?
Yeah, but that may not be that good. Who knows?
Wait till it'll bring them food faster.
They may not be able to capture it as fast.
That's true.
So how come the barnacles are all sort of stretched out in lines?
That's a good question. I don't know, Chris.
A great indicator on the sonars of where the ridge is. If you have
some kind of edge effect.
Do you want me to follow you?
Let me know.
No, come back in. Okay.
Yeah, look at that. They just follow some of the ragged surfaces of the rock.
Some kind of edge effect.
Okay, Asako, I want you to count all those fish for us. Look how many of those things there are. And there's a big old grouper way in the left of the field of view.
She counted 100.
I wouldn't doubt it. And there's some more of those interesting sponges on this ledge. Shirley, you see the thing right in the bottom of the screen.
Yeah.
That was like that
Maybe a
one
white?
that you were describing earlier. Yeah, the white ones. Not the encrusting ones, but that big massive thing.
That had the spicule brushes?
Yeah.
I didn't see it.
I think
was
so.
actually writing something.
Oh, I know.
Typing something, I know.
I know,
you
you're
take
so good.
No, you take your eyes off of the screen for like a nanosecond and something amazing goes by.
I know.
All right, I'm getting all strung out.
Yeah, I might be out of tether there.
This job is a lot harder for the biologist lead.
Do you want to move, co-pilot?
I have a lot of backup, though.
I'll see what.
Yep, that's
I have
true.
a lot
Everybody on the
of
chat
backup.
board.
Yeah, except for Mary
we
Jo
wanna go
and,
to the-
and I
towards
think
that other
Deb had to
waypoint?
drop off.
That's the general plan.
What's the bearing on that?
To
Oh,
the
Mary Jo
waypoint
is still there.
Just about 180, but if the ridge does something else, I think we'd rather follow the ridge.
All right, well let's move.
We
at
really
least 20
appreciate it,
meters at
Mary Jo,
180,
that
and we'll see what
you're
what's out
sticking
there.
with us, even though you
All right,
are actually
I'll get
at
back
a
on.
lab that you are making measurements at in Chicago.
Pilot, you want
And
me to
it's
turn to 180
late
then,
there,
or you
too.
wanna
9 o'clock
look
at
over
night,
there?
You're
10
almost
o'clock
at
at night.
180. I
Oh,
mean,
maybe she's finished for the
yeah,
day.
you can turn to 180. I'll still be in your view.
Yeah. Okay.
20 meters, you think, pilot?
Yep.
Great.
Yeah, we did a time change on the ship last night so that we're making it so it's a little bit easier for
East
those of you
Coast.
on the East Coast to take.
Bridge, ROV nav.
So,
Ready for a move.
although those of you who are
meters,
participating in
bearing
Shorebase have been
180
losing a lot of
degrees,
sleep
0
every
decimal
night,
2
we've only
knots.
lost a couple of hours of sleep in the last few nights.
Right.
We just did 2 time changes and we only lost 2 hours.
Good
whereas
copy, thank you.
most of you on the, I guess probably east of California, I would imagine, are really burning the midnight oil.
And we appreciate it.
We sure do.
Well, we might have a eel sticking out behind that rock. He's just
Which
to the
one?
lower left of center. Not really
Thank you,
sure,
Bridge.
but I think it could be.
Co-pilot, we are on our way
Pilot,
south.
lower left of center. Do you see
you know
an
where
eel
it's at.
sticking out of
Thank
a
you,
rock?
nav.
Lower left of center.
Come on in video.
Yeah.
Very
Yeah.
good.
Good
Wow.
eye, good eye.
All right, I'll try to get a little closer.
Oh, and what's that over on
Oh,
the right?
it's a beauty.
All right.
We'll get over there, take a closer look.
Give you a little bit more leash, but it's about as all I want to give you right now.
Yeah. Come on in video. Oop, it's coming out.
Co-pilot, you should be getting close to
Is it
him.
really further?
Copy that.
I was going back into the chat log, see if I could find this. But I think Asako said earlier that in Japan they eat moray eels, and I thought she said she had like moray jerky. I was trying to find that in the chat
Heavy
log.
survey.
Really?
Yeah.
Everything's pretty good.
Gonna come a little wide.
Yeah, Bruce mentioned earlier.
Snowflake pattern on the body. The bars, the articulated pattern with the white
Turn
blotches
and
that are
look at my
broken up
tether.
into smaller blotches remind me of
Copy
something
that.
called
Co-pilot, we're about halfway done with that
Kidako.
move.
But I'll
thank
have to do some research to find out if that's been reported in the Marianas. It's known from Japan and the Hawaiian Islands.
I got a
And maybe
rock in my
Brian Green,
starboard.
who's on the chat log,
Yep, go ahead.
has an idea from his deep diving.
Yep.
You noticed us.
And Brian just chimed in that he was also thinking of that species, Gymnothorax kidako. So maybe I could find a common name for that species.
Snowflake moray.
Oh, he's coming out. What a beautiful, beautiful beast.
So this is a different species than the one that we saw before. The one that we saw before did not have that the sort of snowflake white blotchy pattern on the body. It was more, I think, barred.
So is there another one that is already called snow? Is that the other one called snowflake moray?
Co-pilot, are we good here for a bit?
Well, there's a shallow water
stay here
species
for a second
called
and let me
snowflake moray
push you
that's
out
more
once we're
white
happy with
than
this guy.
yellow spotted.
good.
It's got a more rounded snout, and you see it in very shallow water, just at snorkeling depths or deeper. It's got a rounded snout, and the teeth in that one are like human molars. It feeds on snails and clams, crushing, and crustaceans, crushing. This eel would have sharp, pointed teeth like people usually think of with moray eels. So the snowflake moray is in a different genus, Echidna, than this one. Okay. Looks like the common name for this, if it is the species we think it is just the Kidako moray. Kidako is a Japanese name. Maybe Asako has an idea about that.
I think Asako might be offline right now.
Well, she just chimed in on the chat room saying,
oh, okay, cool.
The Kidako moray is the edible species that she mentioned in her previous comment.
Right. I was looking for that in the chat log because I think she mentioned it was one that she had kind of as jerky, so dried. How did it taste?
Come out and get the rest of it.
Oh, we might get a Japanese lesson too, because Bruce is asking Asako what Kidako means.
So a couple things about morays. I don't think we saw this last time, but when they're trying to swim from one hole to the next hole, they'll swim sideways. They don't sort of swim up and down. So he's kind of turning sideways, but he's not really, I don't think he's going to, yeah, there he goes. You see, he's swimming sideways, which is pretty interesting, and that may be the reason why he's got those nice, big, long fins. The other thing I don't think you noticed is that I talked about fish eyeballs the other day and about the aphakic aperture, which is the oblong pupil. And I was looking at this eel, and he has the round pupils. And if you look at his eyes, where they're positioned on the head and where you can actually see the lenses in the eyes on that overhead view we're looking at, I don't believe this eel has binocular vision.
Those laser dots are 10 centimeters apart, about 4 inches apart. So you can see it's pretty long. Pretty long. Lots of moray jerky.
Yeah, it looks like it could be half a meter.
Thank you, Pilot.
Maybe more. Yeah, a little bit more. Oh, big boulders up ahead.
Pilot, do you want me to zoom your sonar at all?
Jagged
30 meters.
territory for an ROV.
There's 20. I think that looks good. Co-pilot, you're still at 50.
Yeah, that's good.
So that's 180. Not.
Well, it doesn't seem to be anything there.
It goes downhill. It's
yeah, on my map that should be it. So maybe
maybe we got to go downhill
maybe
a little bit.
More like
Or what do you think?
220, something like that. See how that looks.
Yeah, I got nothing in my sonar. Let's look the other way.
So we are pretty much at the top
Yeah,
of this part of the ridge. And so the pilots are looking around for an interesting place to go a little bit deeper.
Maybe we can go more to this.
Try that.
And see what we can see.
About this heading, you think?
Yep.
Yeah.
All right. I'll do 220.
And there's a shark. Oh, lovely
I'll
thing.
Silver, is that another silver?
Yeah, I think so. Looks very similar to the other one, the other two we've seen on this dive. He's coming right toward us. Look how graceful that guy is. So I understand from Bruce, these guys are more assertive, we will say. I guess we don't look too appetizing.
Yeah.
Nice follow.
A lot more corals down there on the seafloor right in front of us.
Should we get that moving?
Kind of small.
So what did we decide on?
I think we're going to do 220 and maybe just 20 meters.
Yeah, go ahead. Yeah, that's fine.
I'll do that. Bridge ROV nav. Would like another move.
There are some stalked-looking
bearing
things
220 degrees,
that do not have crinoids on top. I wonder if those are just the stalks of the crinoids. Thousands of fish. Tiny fish.
Yeah.
Good copy, thank you.
I can't give you a whole lot more leash, Josh.
Yep, that's fine.
My altitude
Interesting.
is
Now we
16.
are
I
back in the
to,
light
here.
brown
Do a quick
ash
zoom here video.
deposit that we saw deeper on the flank of the seamount.
Thank you, Bridge.
Up here, it's all broken up. Little coral growing out of the top of one of these blocks.
So, I'm going to throw this out, but this is this weird primnoid coral that we saw the other day. And I'm just going to throw this out as a possible option for a second biological collection, because I've never seen a primnoid that looks like this, and I think in the chat it was sort of the same impression.
I see.
Come on out. Come on out.
Pilot, there was a request perhaps to collect that coral because it is
stop
unusual.
the ship move and move them towards me.
Sure.
Video switching out.
Yep. Bridge ROV nav.
So you'd like to sample this watch lead?
Can we come to an easy
perhaps,
stop here?
yes. We're going to just ask folks on shore what they think. And Shirley, what do you think?
Thank you, Bridge.
Yes.
Hey, come on in, video.
Yeah.
So,
okay, we have
was enthusiastically endorsing the collection
I'd like
idea,
to
and
move
Scott
Sirius
had no
towards me.
objections. So we've got 3 people that think this could be worthwhile.
Would that be about
Understood.
180?
Okay.
And you had me on
Pilot.
4.
How far off?
Pilot, you are requested to sample this
15
coral.
would be good.
All right, copy that.
Yeah.
All right.
The coral the target?
Yes.
Okay.
Yeah, and it may be that, I don't know, maybe attached to a very small rock that, so you wouldn't have to try and rip it off from the bottom.
Trying to keep me as much tether as I can.
But we will need the whole thing because it's small.
Well, you got an altitude of 20 now.
Okay.
Bridge ROV nav.
Come down. Copy.
Like to move in a slightly different direction. 15 meters, 180 degrees, 0.2 knots.
Maxim?
Is that where we want to
copy.
be?
Wow. Nice.
Yeah, that'll work.
That's going to put you out in front of me.
Oh, 'cause after this move.
No, he's
Yep,
he's
so we're
stopped.
thinking that might be a
So
Norella.
that's going to put you in front of me. He's going to go 180, right?
That is a beautiful little thing.
Yes, I just called in 180.
Wait, it looks like it has a black stem.
Go ahead, Bridge.
Oh, it's really dark. I don't know. I really
dark.
don't know anything about black corals, but I'm just wondering. Chris would know.
It's not black. It's a
okay.
Frinoid. And they're thinking, our experts are thinking that
okay.
it is...
We'll keep that in mind. Thank you.
Norella.
All right, I'm going to spin around then. We're going to keep this move. All right, co-pilot, can you figure out what box is available?
Our depth is 286 meters.
Or nav?
Yeah, I can do that.
How many samples are we allowed to collect on this dive?
It looks like you could do port
2
out.
and 2
This
typically.
is a biosample, correct?
Yeah, I need a bio box.
Yeah, we could do port outer, or we could do either on starboard. We have 2 different samples in port inner. It looks like the geo sample
and all of these critters are sitting on volcanic ash deposit.
Thinking port outer.
Yep. Do you see where he... Oh, there he
yes,
is.
survey. Got you.
All right, port bios prepared for opening.
Yeah.
Sure, will do. Watch lead, Nav.
Yeah, go ahead, Nav.
Hey, are you concerned at all about the sample in the port inner box, that piece of coral floating out, if we open that box up again?
We'll go to starboard then.
That's the one that has the crinoids on it.
We'll go to starboard.
Yeah, so what are you thinking? Don't open that one, go to the other side.
Pilot's telling me we can go to starboard if you'd prefer that, which has nothing in it right now.
Co-pilot,
Yeah, my
let's
preference
get
is to
starboard
go to
ready.
starboard.
All right,
Got another moray eel there.
Pilot, they'd like to go
Different
starboard.
species.
And there's an eel poking its head out of the rocks right in the center of the left side of the screen. It is wondering what is going on.
Almost done moving, navigator?
Yes, we're about done with that move.
Excellent. All right. Starboard is ready to go.
Make sure that the port's not going to open.
Yeah.
And you can see the manipulator coming into view.
Got me Zeus over here.
The fingers, or the silver part of the manipulator, are about 9 inches long for scale.
Guys,
So when we collect these samples, they are deposited in museums, archival repositories, so that anyone who wants to request to use them may. And the biological samples are going to the Smithsonian Institution. A small piece of each biological sample is being subsampled for molecular analyses. Those samples are going to Ocean Genome Legacy, another OER partner at Northeastern University. And the biological, geological samples, the rock samples, are going to Oregon State University.
It's a little far in your claw right now. See the mini Zeus.
Wowzer.
Really well done there, pilot.
Hey, tilt up on main Zeus.
Although it is quite a small specimen,
That's
our
okay.
pilot
Wow.
was able to
Another
pick
great
it up.
job.
Yeah.
That was
Pick it up,
amazing
yeah.
for a coral that small.
Yeah.
You are being heavily congratulated,
Yeah.
pilot.
See the
for sure.
crinoid swimming
Yeah.
in the background.
Come on out.
Kudos
on
from around the world.
All right. Clear on the box.
Drawer coming open.
Yep.
Oh yeah.
Yeah.
There
Yeah,
is
it's coming open.
there is a crinoid walking across the bottom. If
Yep.
you folks
Yep, well.
just below the dark
Like
outcrop,
at about 8 o'clock. Yeah, about 8
about 2
o'clock. 2
o'clock.
o'clock. Yeah, about 2 o'clock.
and they do that. Although the ones that were on the coral that we picked up earlier decided to take a ride with the coral they were on.
Okay.
Can you bring up mini Zeus on the second cam?
Right monitor?
Yep. Tilt it down.
We have a lot of different cameras and lights on the D2. Beautiful.
All right.
Okay, let's see, make sure it's not stuck.
No, I think I saw it fall off.
No,
I did not.
no, it's on there. Okay, that's all right, that's all right, that's all right.
It's very difficult to flick a mechanical arm, but you can tap it on the side of a bio box.
Nice, nice. Nice, really nice.
Really well done.
Really, really nice.
Okay, clear.
So the pilots are sliding that bio box back underneath the vehicle. So now that bio box is closed and nothing will float out when we bring the ROV back to the surface.
How many square up your minis is?
Yep. Okay.
Large grouper in the background.
What's happening here?
There's a grouper up at the top of the
Take
screen.
my HD back.
Plenty of small fry for him to eat in
That's
this
you.
area.
Was that crinoid still
Give you your
visible,
cameras
the one
back
that was moving along?
I'm not actually
All right.
sure, but
No,
I'll
he swam
ask.
off.
We got the
to right.
Let's see.
waypoint nav.
To the
Yes, I
left
have
or
it
to the
drawn.
right?
I'm sorry, yes.
The one that you saw
Don't have
earlier.
drones.
You're making me feel a whole lot better.
Is that it's nice.
I saw a real big grouper swim around behind
Nice
the rock
collection
in the
there.
upper left, right.
Thank you.
That was a tricky one.
But the crinoid was
are
crawling
the-
along
Those
toward
are the
the
best
right.
ones.
Was it a stalked? Oh, no, it was that one.
There's the
I see.
And
crinoid
there it
landing on
is.
the right.
Immaculate.
Settling down now.
Yeah.
That's really interesting.
Come on in, video.
So it must have gone up in the water column a little bit
Looking for something to land on.
and then settled back down.
Something to land on and attach to. It looks like it's right next to a brittle
He's
star.
been exercised enough.
Okay.
Co-pilot now.
No, it's
Oh,
next to a
squat
squat lobster.
Do you know if this spreadsheet is working correctly with the new time zone? It
So
looks
its little
right.
Cirri are the white
be good.
hair-like
Yeah,
things
1622
underneath
off
the
bottom,
crinoid
so
that it's going to grip
yeah,
the
that
rock
sounds right.
with, right?
And these look like they are the non-prehensile ones.
Correct.
Yeah, for a 1630 local
Yes.
recovery.
So that coral that we just collected, Narella, it's a large genus, a lot of species. Scott France tells us that there are at least 50 described species of Narella. Looks like it's a young colony of something that we may typically see larger.
Video's clear.
Okay, what is happening over here?
Well, the squat
There's
lobster
some interaction.
has grabbed a portion of the arm of the crinoid that just landed there. I don't know what he's going to do with it.
Or maybe vice versa. Maybe the crinoid's grabbing the squat lobster.
Well, the squat lobster did open its claw and take hold of the arm. But whether the crinoid might like to try to eat the squat lobster, I have no idea.
I don't think so.
Looks like the squat lobster has
one interesting thing you'll note about the squat lobster is how long his pincers are.
Yeah.
Yes, and indeed.
That's characteristic of some types of these. I've got to remember which is which, but look at how large those claws are.
They really are. Yeah, the first thing it did when it felt the crinoid is it backed into its little crevice. And then it came out and grabbed the arm.
So, yesterday we saw an octopus that was going along the bottom, and we think eating.
I didn't know what in the world it
would
could
eat.
But sure enough, it settled down on top of a squat lobster, and then it went on, and there was no squat lobster left.
So now we're starting to see corals again. Guccisamia, another grouper in the background. Lots of barnacles. All that white along the on these rocks are all barnacles.
Groupers are all hanging on the backside of that cliff.
Well, this has been a fascinating dive for a bunch of reasons.
Another grouper straight ahead. Two of them.
Yeah, we could probably put in another move at 180. Start heading down towards that waypoint.
Okay. Yeah, we are due north of it, about 80 meters away, so we have plenty of time.
So we have about an hour and 45 minutes of bottom time left.
All right, I'll have us do another 20 meters. Do you want to go slower? We've been moving at point 2.
Yeah. Okay.
There must be a fish we have not yet identified.
Okay, come on in, Roland.
Lots of them.
Is that a siphonophore?
Okay,
is that the right way to pronounce that?
Be 20
siphonophore, yeah.
20 meters, 180.
Yep.
Sounds good. Thank you, Nav.
It looks like it's got sort of a cup shape on the
like a ball at the top.
Right.
Bridge, ROV nav. We're ready for another move. 20 meters, 180 degrees, 0.2 knots. Good copy, thank you.
Pulling out.
That definitely falls off to the east. Thank you, Bridge.
Another grouper. Yeah, Scott, I think it was a siphonophore also. Okay, that grouper is an 8-bar grouper that we saw just a few seconds ago. Mary Jo is weighing in on the geology. Yellow tan ash vugs as before. Broken lithified ash blocks are here, yellow tan interspersed with dark material.
Right.
So, pilot, it looks like this is a little bit downhill, but I think we're still on top of the ridge. Do you agree?
Yeah, that's fine.
Okay.
The surface on the right has this sort of pockmarked character that you see sometimes
Yellow coral.
on beach rock in Hawaii.
Diagonally down right.
But the beach rock is made of carbonate. And we did get a piece of this stuff.
Come on in.
And we'll check it out for you, Mary Jo, but I'm pretty sure this is ash. The nature of the layering that we saw suggests it's very likely ash deposits. We have a real interesting looking little coral here. It's sort of yellow where the polyps are.
Looks like the Parasus. Is that what we called it before? Parasus?
This is so delicate.
Parasus.
What's that in the background?
Oh, and look, there's one of those sponges
Yes.
that you like in the cracks. In
Yep,
the hole in the
yep.
rock beneath it.
Hexactinellid sponge with zoanthids.
Hexactinellid.
So you're at the edge of my
when you're happy.
green pilot. We're moving. It's only 20 meter move though.
Yeah, we have about 9 more meters. It looks like you might get ahead of you a little bit.
It's not that big of a move, so you'll be all right. But just letting you know.
Lots of coral.
Yep. Seeing a lot of those. Yep. So now we see where we saw. Lots of corals. I think. Are these all the Parasus?
Oh, and there is
beautiful, lovely yellow crinoid.
Just when you think something looks like a fern, it swims away.
And this little crinoid, you can see the white, very thin cirri, kind of like feet, kind of like anchors, right below the body of the crinoid. That's what it uses to grip onto the rocks. These guys are filter feeders, right? Not really filter, but they grab stuff out of the water.
Suspension.
Looks like
suspension feeders.
There's another one. Oh, there's a group of them. I think they should get a room.
Oh, they're all hanging on that coral.
Are they?
Yeah,
See a coral in there?
Coral underneath.
Take your word for it. I don't see it there. In the background, there's a bunch of them on another coral.
What I want to see is another
negative 99, so I'm looking back at you just a little bit.
Our move has stopped.
Now there is
should be okay.
There is something dark that looks a little different from the other corals in the middle of the screen.
It's a bunch of crinoids on top of a coral.
Oh, is it? Yeah, you're right. That's cool. There's a yellow crinoid and a dark crinoid. 2 different types on this coral. Does anybody on shore have an idea what the different coloration might indicate?
Of the crinoids?
Yeah, and the crinoids.
If they're different species?
Different species. They look real similar in terms of their arms.
I'm just looking for something that Scott was pointing out, a stalked sponge somewhere, but I don't see it. There's something, looks like a something cylindrical in the background.
Get over to the cylinder. Can we do a quick zoom on this white guy in the middle? That's crinoid.
Survey 1 nav.
Right, coming up.
That was a closed up anemone, the cylinder thing.
Wind is picking up a bit to our port. Should I talk to the bridge about this, or will they ask if they want to change heading?
Oh, there's an
Watch
anemone.
the pilot.
Go ahead.
Yep, you're zooming into it.
It's fine for us, so I'll hold off unless they call it.
That cylindrical critter looks like an anemone to me. We'll see.
I'm not
Maybe
going
it's
to.
a
I'm not
sponge.
going to say anything until we get closer. And looks like you're right.
Wow, it looks
Looks
like
you're
a
right.
closed one of those
Yeah,
things that we
yeah,
saw.
yeah.
This is a closed-up Isaac Nernus anemone.
Wow.
Now, is it closed because it's consumed something?
I don't know. Good question.
Chris, do you think maybe it's eating whatever it recently caught?
I have no clue why it do that. No idea.
So why else
What
would
about
it close up? Why do they, is there anything that makes them close up that isn't because they're eating?
Well, eating would be one reason. They caught something and they're trapping it so they can digest it. That's definitely a possibility. It's not going to be like that anemone, that weird anemone that we saw the other night.
Tara, go ahead. Did you want to say something?
Hey, come on up.
Yeah, we had a little more chatter in the room here, so I was just waiting a bit. But the anemone that we saw yesterday, the weird one with all the nematocyst batteries all over the stalk of that one, that's an anemone that comes out at night and then closes up during the daytime. And we know this because I remembered that we collected a live one many years ago and gave it to the Waikiki Aquarium, and they kept it alive for several weeks, and they saw that it would come out at night and close during the day.
Go ahead.
And so that's the reason that one was closed up. But we have seen these anemones out during the day. That's the first closed-up one we've seen. So it's not that. There has to be another reason why that one's closed.
Well, maybe something was coming along and trying to eat it, and so it retracted its tentacles.
Yeah, it's possible. And what I sent Scott an email this morning because I was thinking about that anemone from yesterday and about all the nematocyst batteries on the stalk, which are quite unusual. And it just dawned on me driving home last night that when that animal's closed up, which is how we saw it, basically it's totally vulnerable to predation, and I don't think it's got a very thick skin. And so I think it's got all of those nematocyst batteries to protect itself during the day while it's all closed up, and it doesn't have any tentacles to try to protect itself.
Thank you, Chris. We're zooming in on very feathery-looking little
pinnate
coral
octocoral
with
a crinoid at the top.
At the very top, yeah.
And beautiful blue fish. So that's a primnoid and possibly Caligorgia. And just in front of this one is another one.
We're being followed
so
by
I'm not
beavers.
positive that this is Caligorgia, but it has alternating branches, so it could be something like Caligorgia gilberti if it is Caligorgia. One thing you look at is whether the branches are coming out opposite of each other or alternate, and that one was alternate.
Mary Jo's commenting that the ash layer seems to be blockier here.
Yeah, I agree. But still layered, and what's happening is that the individual layers seem to be spalling off. This often happens when you have graded bedding in individual ash layers, so that you have coarser grain fragments on the bottom of an individual layer and finer ones up at the top. So the coarser layer often acts as kind of a ball-bearing layer that can permit things to break off.
Is that another
along
primnoid? That looks different.
That's a really delicate-looking
Just the
coral.
side view of the same thing. I
Is
think.
it? Is it? So it's another primnoid?
Yeah, I believe so.
I just can't tell with the branching on that one.
Excellent.
All right.
Redfish in the hole there.
Come on in.
More barnacles.
These holes that we see in the ash beds are so round. I'm wondering if something has bored their way into these things.
Nudibranch or holothurian?
So is
What is
that?
this little white guy? Is that a holothurian of some sort?
No, that looks like it might be either a looks like a nudibranch.
nudibranch.
Yeah, it looks like a nudibranch.
Okay.
Isn't that pretty?
That's very cute.
And I'm looking over to the left and I'm seeing a different kind of octocoral than we've seen. Oh, that's nice.
Oh yeah, that coral is real pretty too in the very upper left-hand corner.
I'm going to get Scott's opinion on this, but this looks like that really strange plexaurid that we collected in Hawaii that nobody knew what it was, and I think we settled in on Swiftia. That's that red-stemmed yellow one, and maybe Asako has seen this thing and knows what it is. Seems awfully shallow for it compared to the one we collected in Hawaii.
Looks like he's chasing down that little fiddler crab.
You wonder if he's chasing that fiddler crab, huh?
I mean, hermit crab? Hermit crab.
I think I just saw a pink
yeah.
in the upper left. I think that's what the sign is talking about.
He's just moving along the bottom.
Yeah, I wonder, it could be eating the worms from the worm tubes too. Maybe.
Grazing along.
Nav, co-pilot.
Go ahead, Nav.
Do you know what the story is with our track lines on HiPAC? It's like we have.
I guess we're going to go have a look at that coral.
Yes, we are. Yes.
Okay.
Sirius is there.
It's just.
It's
yeah,
just
I see it.
very, very close together as we've been sitting for a bit.
Watch lead, pilot.
Go ahead.
Yeah, if we can just get some good lighting on that, some good video. Thanks.
You want to adjust the lower right swing arm, pilot?
Absolutely.
Might not be able to get a bunch of light on it.
What would you like?
Light on this coral.
Copy that. See what I can do for you.
I didn't hear what Chris said earlier about which one he thought this was, but it doesn't look to me like Swiftia, if that's what he was suggesting.
Yeah,
some kind of plexaurid.
Yeah.
Yeah, when we got a better view of it, I changed on it. I would agree it doesn't look quite like the same one that we collected in much deeper water in Hawaii.
Yeah.
But I'm hoping that Asako knows this one.
It doesn't look like she does right now. I would say this would be in the old family Paramaricia day, which doesn't exist anymore. It's been subsumed in the Plexauridae, but it's, one of those Paramaricia-like groups.
On top of the rock.
So while we're looking at this, I can tell you that the colors that you see on these octocorals come from both sclerites, which are calcium carbonate microscopic elements that are embedded in the tissue, and from pigments that are in the tissue. And so one of the ways you get the different colors is This may have sclerites that are pink, and the polyps may have pigment that is yellow in the tissue. It's also possible that you could have different color sclerites, and so on.
Thanks, Scott.
Did it get any better? I'm bringing it in pretty far here.
Yeah, I don't remember seeing any sclerites that are this color. I recall seeing some that are like bright purple, but I don't know if I've ever seen any that are this kind of salmon pink color.
All the way around the other side, which we can't fit inside that crack.
I'll bring it back out. I'll bring it back up.
And so the way that we use spicules to identify sponges, are sclerites used to help identify octocorals?
Absolutely. That is the prime characteristic used to identify the species, and it's one of the reasons why it becomes so difficult to identify them in these videos. If we knew, for example, in Hawaii, above 500 meters or above 1,000 meters, where Chris and his program has done an awful lot of collecting. They have a very good sense of what's there because they've been able to collect a lot of corals, and then they look at them microscopically at the sclerites and some other characteristics, and then relate that back to the images they've seen. Here, where we're exploring new areas, we're making a lot of guesses based on what we've seen from other places, but it's purely a guess when we try to say what species it is because we don't have that look at the microscopic elements.
Something else in that cave, an eel, maybe a fish fin.
Okay, anything else, Watchley?
Nope, that's good. Okay, we are at about 285 meters, 286 meters on Supply Reef, Marianas Trench National Monument. We are coming to you live from the NOAA ship Okeanos Explorer, which is America's ship for ocean exploration. This mission is designed and supported by NOAA's Office of Ocean Exploration and Research, OER. The ship is operated by NOAA's Office of Marine and Aviation Operations, and the ROV is operated by a NOAA partner, the Global Foundation for Ocean Exploration. Other NOAA partners involved in this are NOAA's National Marine Fisheries, NOAA's Marine National Monuments Program, NOAA's Deep Sea Coral Research and Technology Program, and also NOAA's National Center for Environmental Information. This partnership is what makes this all possible. We collect a limited number of samples and a lot of data and a lot of images, and all of these data, samples, images will be available within 60 to 90 days to anyone who you just submit a request, and you can find out how to do that on the oceanexplorer.noaa.gov website. But it makes it a quite unique type of expedition. Most of the expeditions that I've been involved in have been single scientist or just a few scientists led. We are all out to sea together on one ship, and we have a couple of years to use our data and samples before they're made publicly available, and sometimes they're never made publicly available, although that's changing. This is a lot different, so the idea here is to engage as many explorers and scientists as possible so we can... I knew this was going to happen. I just knew it. I just knew it. I just knew it.
We're just looking at that pink coral on the left.
Yeah, we're looking at a slit shell right now. I just knew this was going to happen. Really one of the target